Welcome to this lecture on Parasitic Platyhelminthes. Imagine hosting a dinner party where a guest sneaks in, eats your food, and never leaves, even dismantling your house for armor. Today, we will explore these ultimate biological stowaways, understanding their unique biology, the devastating diseases they cause, and the crucial mathematics we use to diagnose and treat them. Now that we have introduced our uninvited guests, let us look at their biology. In formal terms, parasitic platyhelminthes are dorsoventrally flattened, bilaterally symmetrical, acolomate invertebrates. Because they lack a true body cavity, known as a column, and have no complex circulatory system, their flat shape is actually an evolutionary masterpiece. This unique morphology allows them to absorb nutrients and oxygen directly from the host through their outer surface, which is called the tegument. They are perfectly adapted to survive in the harsh, acidic, and immune-active environments inside their hosts. Building on their unique anatomy, we classify these flatworms into two major medically important groups. First, we have the class Trematoda, which includes flukes like schistosoma and the giant intestinal fluke, Fasciolopsis busci. Second, we have the class Cestoda, comprising tapeworms, such as Taniosolium. These parasites do not just jump from person to person. They have incredibly complex life cycles. They typically require one or more intermediate hosts, such as snails or pigs, to develop. Only after this journey do they reach sexual maturity in their definitive host, which is very often a human being. Understanding these life cycles brings us to a critical question. Why dedicate an entire field of study to them? The reality is that parasitic platyhelminthes cause some of the most devastating neglected tropical diseases on the planet. For example, blood flukes cause schistosomiasis, which affects over 200 million people, leading to severe organ damage. Similarly, the pork tapeworm can cause neurocystocercosis, a leading cause of preventable epilepsy in developing nations. Without studying these parasites deeply, we could not develop life-saving drugs, design public health interventions, or protect agricultural economies from massive livestock losses. Knowing the severe impact of these diseases, how do we actually detect them in a patient? When a doctor suspects a flatworm infection, they cannot simply look inside the intestines to count the worms. Instead, they must search for microscopic eggs that the adult worms shed into the patient's feces or urine. However, merely finding an egg is not enough to treat the patient effectively. We need to know exactly how severe the infection is to prescribe the correct dosage of medication. This is where parasitologists turn to standardized mathematical calculations to determine the infection intensity. To quantify this severity, we use a fundamental diagnostic equation. Conceptually, this formula scales a microscopic observation from a tiny sample up to a standardized macroscopic metric. In this equation, I represents the infection intensity, expressed in eggs per gram. C stands for the raw count of parasite eggs observed under the microscope. W represents the weight of the stool sample used, measured strictly in milligrams. The number 1,000 is a conversion factor to scale milligrams up to a full gram. If the egg count C increases while the sample weight W remains constant, the overall infection intensity I will increase, indicating a more severe parasitic burden. Now that we have our formula, let us put it into practice with a clinical scenario. A patient presents with symptoms of an intestinal fluke infection. The lab prepares a smear using exactly 41.7 milligrams of stool and counts 45 Fasciolopsis buski eggs. We need to find the standardized intensity. First, we substitute our known values, replacing C with 45 and W with 41.7. Next, we divide the egg count by the weight, yielding approximately 1.079 eggs per single milligram. Finally, multiplying by 1,000 gives us 1,079 eggs per gram. This quantitative result tells the doctor the patient has a moderate infection, guiding the correct dose of praziquantel. Having calculated the infection intensity, we must realize it only tells half the story. Knowing the number of eggs per gram is incredibly useful, but it only reflects the reproductive output of the parasites. It does not tell us the actual number of adult worms living inside the patient. To truly understand the physical toll on the host's body, we need to estimate the total adult worm burden. We do this by working backward from the egg count, using the known biological reproductive rate of the specific parasite. This mathematical estimation is absolutely vital for predicting the risk of severe complications, such as intestinal blockage or organ rupture. To unveil this hidden population, we use the population estimation formula. Conceptually, we are stripping away the reproductive multiplication factor to reveal the base population size. Here, B represents the estimated worm burden, which is the total number of adult parasites. I is the infection intensity we just calculated. P represents the per-worm fecundity constant, meaning the average number of eggs a single adult worm contributes to 1 gram of feces. Interestingly, if the infection intensity is very high, but the fecundity constant P is also very high, the actual worm burden might be lower than expected, because just a few highly reproductive worms can produce a massive number of eggs. Let us apply this to our previous patient to find out how many worms are actually inside them. We already established the infection intensity is 1,079 eggs per gram. Biological research tells us the fecundity constant for Fasciolopsis buski is approximately 179 eggs per gram. We substitute these values into our formula, dividing 1,079 by 120. This gives us approximately 8.99, which we round to a worm burden of 9. This means there are an estimated 9 massive adult giant intestinal flukes feeding inside the patient, fully justifying immediate medical intervention. Now that we have mathematically quantified an infection, let us visualize how these parasites spread by looking at the life cycle of the pork tapeworm, Taenia solium. At the top, we see the human, the definitive host, harboring the adult tapeworm in the small intestine. Following the arrows clockwise, the human excretes feces containing eggs, which contaminate vegetation. At the bottom, the pig, acting as the intermediate host, ingests these eggs. Inside the pig, oncospheres hatch and migrate to the muscle tissue, forming cysticerci. Finally, the cycle completes when a human ingests this infected, undercooked pork. This diagram perfectly illustrates the complex, multi-host journey these parasites undertake. To conclude our lecture, let us review the most important ideas. First, parasitic platyhelminthes are highly adapted, flattened worms that cause significant global morbidity through their complex, multi-host life cycles. Second, diagnosing these infections requires rigorous mathematical quantification. We use the infection intensity formula to measure disease severity based on eggs per gram of feces. Third, we use the worm burden formula, to estimate the actual hidden population of adult parasites inside the host. By mastering both the biology of these fascinating stowaways and the mathematics used to track them, medical professionals can accurately gauge disease severity and save lives. You now have a solid foundation in medical parasitology.